Hi everybody, this is Fatima Al-Ahmed and my partner Ghada Al-Huwaidi. Today we're gonna present about a paper was published in 2017. That paper was about improvement of the glycoproteomics toolbox with the discovery of ionic C-terminal cleavage specificity of avavestin for N-glycosylated asparagine. And the outline will be introduction, objectives, materials and method, result and discussions, and conclusion. Introduction. Proteins are the most important functional component of living organisms. Protein glycosylation is being used in several key biology functions. Apparent glycosylation is associated with the various cancers. Many inflammatory and infectious diseases are linked with a glycosylation variations. Due to defect in glycosylation are genetics and metabolic disorder are common nowadays. These defects in glycosylation are called congenital disorder of glycosylation. The study of the mechanism of a protein glycosylation will help a lot in the future for the prevention of various diseases and disorders. There are two main forms of protein glycosylation, N-linked and O-linked glycosylation. N-glycans are linked to the amino group of asparagine according to a specific sequence. O-linked glycosylation is attachment of a sugar molecule to the oxygen atom of a serine or a threonine residues in a protein. Comparison of avavestin and peptidyl ASP metalloendopeptidase. The protein sequence of avavestin Q47899 and peptidyl ASP metalloendopeptidase Q9R4G4 are from Uniport. Both sequences were compared using the protein blast tool. Use the protein blast tool from the National Center for Biology Information. There is a free online tool. Sequences with a similarity are illustrated in the lower box. The first sequence has a similarity of 11 out of 49 amino acids, approximately 23%. The second sequence has a similarity of 4 out of 9 amino acids, 44%. Possible conservative amino acid chains are indicated with a positive charge. The field of site-specific glycoprotein analysis method has grown tremendously in the past. It emphasized the importance of protein glycosylation analysis with respect to micro and macro heterogeneity. Site-specific glycoproteomics in the analysis of specifically or unspecifically digested glycoproteins via liquid chromatography combined with a mass spectrometry. A glycopeptide enrichment can also be performed prior to analysis, for example by a hydrophilic interaction chromatography. Investigation of a new tool for targeted N-glycoprotein digestion to overcome common problem in glycoproteomics, like a large N-glycopeptide with too many amino acids for a proper LCMS measurement, like a multiple N glycosylation sites with one in glycopeptide. Materials and methods. Chemical used. Proteolytic digestion via filter, aided sample preparation, FASP, LCMS to MS measurement, and data analysis. Chemical used. These chemicals were used during experiment. Protein, bovine serum albumin, BSA, A3912 and 100G, and electrotransferrin. Enzyme used for digestion were trypsin and endoproteinase, ASPN. All solvent for LC were MS grade, and all buffer and solution were prepared with a deionized and buffer water DH2 using a molecule water purification system. For LCMS solvent was further buffered using the LC pack polisher from Merck Millipore. Manually annotated in a glycopeptide sequence of a human electrotransfer HLTF from MS to MS spectra of a nano RP LCESI OTMS to HCD. Measurements after sequential digestion of HLTF with atropine and favastin. CAM, a carbamidomethylation. MSO, methionine oxidation. Proteolytic digestion via FASP. BSA and HLTF were digested proteolytically using a modified version of a filter aided sample preparation FASP method. 100 micrograms of each protein were applied to a filter unit. Samples were treated with a urea buffer, followed by reduction with dithoerythritol and alkylation with isocyanates in 50 millimole ammonium bicarbonate, and filter unit was washed 3 times with a urea buffer and 3 times with ABC buffer. Proteins were digested with trypsin using an enzyme protein ratio of 1:30 weight to weight and incubated overnight at 37 degrees and 350 RPM using a temperature controlled incubator. Digests were collected by centrifugation. The flow through was kept along with the digest in order to be dried by vacuum centrifugation. 20 micropeptides were reconstituted in 20 microliter 1 times ASPN reaction buffer and ASPN flavastin was added enzyme protein ratio 1:20 to the peptide solution and incubated overnight at 37 degrees. The flow through a tripartite digest of PSA and HLTF, as well as the sequential digest of PSA and HLTF with trypsin and ASPN flavastin, was dried by vacuum centrifugation and reconstituted in 0.1% trifluoroacetic acid prior to LCMS2MS measurement. LCMS2MS measurement. The LC system was ultimate 3000 rapid separation LC system from Thermo Fisher Scientific samples around or approximately 500 were loaded isocritically on a trap column via 100% loading buffer A, 39% DH2, 2% ACN, 0.05 TFA with a flow rate of 7 microliter with the first 5 minutes. The loaded trap column was switched in line with aspiration column with a nano flow rate of 0.3 microliter per minute of 4% nano buffer P trifluoroethanol TFE, 8% ASN and 0.1% formic acid Fa and nano buffer A. The separation was performed by a multi-step binary nano A to B gradient 4 to 55% nano buffer P till 80 minutes, 55 to 90% nano buffer B till 100 minutes, 90% nano buffer B till 110 minutes, and 4% nano buffer B from 110 to 150 minutes.
Result and discussions. They present a newly identified cleavage specificity of vastatin, which up to now was only reported to cleavage peptide bonds N-terminal to aspartic acid residues. In contrast, they could not confirm this N-terminal specificity of vastatin for aspartic acid. However, for the first time, they show a unique cleavage specificity of vastatin toward the C-terminus of N-glycosylated asparagine residues. Implemented in an N-glycoproteomic workflow, the use of vastatin can thus not only render data analysis much easier, it can also significantly increase the confidence of mass spectrometry-based N-glycoproteomic analysis. They demonstrate this newly discovered specificity of vastatin by liquid chromatography mass spectrometry analysis of complex type glycosylated human lactotransferrin and bovine serum albumin peptide and N-glycopeptide that were generated by trypsin and vastatin digestion. Following to this work, they conclude further elucidation of this efficiency, specificity, and mode of action of vastatin is needed, but they believe that their discovery has great potential to facilitate and improve the characterization of N-glycoproteomics. In this figure for site N497, the fragment ion spectrum of the N-glycopeptide sequence TAGWNIPM star GLLFN497 with the N-linked glycan and the corresponding precursor ion 1184.1659. The star indicates the carbamino methylation of methionine. In this figure for site N156, the fragment ion spectrum of N-glycopeptide sequence with the N-linked glycan and the corresponding precursor ion 1321.2479 is shown. N-glycan structures and oxonium ions are illustrated. The B-ion series are highlighted in green, the Y-ion series in red, and the B-ions are in blue. The isotopic patterns of the precursor's are shown at the upper right corner. Specificity of flavostacine for the C-terminus of N-glycosylated asparagine, the amino acid sequence of the tryptic N-glycopeptide of helicase-like transcription factor with N-glycan series linked to the N-glycosylation site N156 is shown. The scissor illustrate trypsin. The shield stands for cleavage inhibition of trypsin due to the proline. Flavostacine is symbolized by a pig and its specific C-terminal cleavage of N-glycosylated asparagine, as well as its M-specific N-terminal cleavage are shown. To conclude, flavostacine shows a clear specificity for the C-terminus of glycosylated asparagine N156 and N497 in helicase-like transcription factor. Due to the presence of multiple N-glycosylation sites and the cell will describe complex type N-glycan structures, the helicase-like transcription factor is a very suitable glycoprotein to demonstrate this newly found specificity of flavostacine. We could not verify the claimed specificity of flavostacine for the N-terminus of aspartic acid, neither for helicase-like transcription factor nor for PSA. However, we could demonstrate that the sequential combination of trypsin and flavostacine for protein digestion successfully cleaves the M-glycoprotein in helicase-like transcription factor, involved and notable in glycopeptide sequences. Interestingly, in contrast to unspecific digestion strategies using proteinase K or ponase, flavostacine works as an glycospecific proteolytic enzyme, and this probability improves data quality as well as data analysis and therefore facilitates in glycoproteomics significantly. However, the unspecific cleavage due to the flavostacine at the N-terminus results in the distribution of redundant N-glycopeptide signals with peptide mayors of different length. Overall, this finding improves the glycoproteomics toolbox and helps to overcome common problems in N-glycoproteomics. For example, the presence of too large N-glycopeptide with too many amino acids and/or too many N-glycosylation sites for proper liquid chromatography mass spectrometry analysis. Other glycoproteins, as well as for more complex glycoprotein mixtures, and in particular for other types of glycosylation, the use of flavostacine will already be beneficial for glycoscience now as it allows researchers to dig faster and deeper into N-glycoproteomics.